back to Now I Know, and today we are talking about telomere and telomerase. The moment we hear telomere, we know that it has something to do with the aging. So that's what we are going to see today, what exactly is telomere and what is the function of telomerase. So let's begin. So let's begin with telomere. Telomere from Greek, it means telos, meaning end, and meros, meaning part. That means it is the end part of the chromosome. Okay, telomere is present in the end part of the chromosome. And what exactly is this telomere? When I say telomere, what does this mean? Telomere is a repetitive sequence that is present in the ends of chromosome. Say for example in humans, it is a G-rich repetitive sequence, that is TTAGGG. This sequence is repeated for hundreds to thousand times in the ends of human chromosome, and that makes the telomeric region for human. Now the function of telomere is it prevents losing of essential genes from chromosome during replication. It prevents fusion of the chromosome end. Maybe this chromosome end can fuse with some other chromosome end. It prevents that, and it protects these ends of chromosome from degradation. Now of course I have listed over here, but how exactly telomere does all this function? That's what we are going to see now. So to understand the telomere, let's start with the eukaryotic replication. We are not seeing the replication in detail. What happens in eukaryotic replication? We know that replication would take place from 5' to 3' direction. And it needs, the polymerase enzyme needs RNA primer, because that gives a free 3' OH end, and that's how it continues with the synthesis. Now I have not shown here leading and lagging strand. Remember, both the strands are not continuous. One is continuous, one is discontinuous. So we have 5' to 3' synthesis taking place. You have 3' OH end, which is provided by the RNA primer on both the strands. Now once the replication is over, we know that these primers will be removed. See, once the primers are removed, you have a gap over here. But now how will this gap be filled? Because there is no free 3' OH and there is no primer available. So polymerase enzyme cannot fill in this gap. So you are losing a part of your chromosome. You are losing some important genes. What if these genes are really important? They code for some specific proteins, or gene products are really required in the cell. So you're losing that particular portion. Now when this cell will divide next time, again the same problem will come, and with few more genes, you're going to lose some more genes. So every time your cell divides, because you don't have a 3' OH end when it comes in the extreme end of the chromosome, you're losing a bit of your genes every time. So your chromosome becomes shorter and shorter every time cell divides. That is not feasible. So this end replication problem, which is called as end replication problem, is solved by presence of telomere. Let's see how. This is where we stop. We don't have 3' OH end to fill in this gap. So every time the cell divides, we are going to lose our some of our genes. So chromosome becomes shorter and shorter. To solve this problem, we have telomerase enzyme. Okay, and the speciality of telomerase enzyme is it contains RNA template of its own. So if this is my telomerase enzyme, this is just the schematic representation. This is not how it looks. It contains its own RNA template in it, and what it does is, using this template, it is going to extend the 3' end of the chromosome. So this enzyme, using its RNA template, will extend the 3' end of the chromosome. And what these bases are? This is nothing but the telomere region. So using this RNase template, the telomerase enzyme will extend the 3' end of the chromosome. Once it extends this much portion, it will again slide back and extend it some more. It will go on for a few more times, and it will extend the 3' end of the chromosome with the repetitive sequence, and that is nothing but the telomeric region. It uses RNA, information stored in RNA template, and synthesize DNA. So it's kind of a reverse transcription taking place over here. Synthesis of DNA from RNA. Right? So telomerase enzyme synthesize your telomeric sequence at the 3' end using its own RNA template. Now your chromosome has got telomeric region. So now when the replication takes place, what happens? Your primer is bound here at the 5' end, and the extension is taking place because you have a free 3' OH end. And at the end of replication, the RNA primer has to be removed. It will be removed. You have the patch which is missing now. It is the telomeric region actually. And as of now, what we know is telomeric region is non-coding part. Okay, it does not code for anything. So instead of losing your coding gene from the chromosome, you are actually losing the telomeric region. So that's how it helps or prevents loss of any important gene every time the cell divides, right? You are losing a part of telomere now every time the cell divides, and that's why after every cell division the telomere becomes shorter and shorter, and once it reaches to a critical length, then the cell can no more divide, and that's how the telomere length is related to aging. When a cell is new, a fresh cell, it has a very long telomere, but every time the cell divides, you tend to lose few base pairs of telomeric region, and as the cell grow old, the telomere length becomes shorter and shorter. Also, when the replication is over, you see this, there is a overhang over here. There is a single strand overhang over here. What this telomere single strand does is, it will fold itself back and tuck itself in the complementary double strand and make a loop kind of a structure. It will not stay as a single strand. The telomere will fold itself back and tuck itself in the complementary sequence and form a loop. And because of that, what will happen is, suppose if this thing doesn't take place, what the cell will think that there is a damage because there is one single strand present in the chromosome. That can be misunderstood by the repair mechanism, thinking that the cell is damaged, and they will come and try repair it. That's how it also prevents the chromosome ends from degradation, because it is going to loop itself back. So how it prevents fusion of chromosome ends? It is because if the telomere does not loop itself, it does not fold itself, if it is still in the single strand, this can tend to base pair with the single strand of the other chromosome end, or maybe the same chromosome end, because it is in the single stranded form. So because it has the folding back and loop formation, it will prevent the fusion of chromosome end, right? So it's very clear now how it helps in the prevention of loss of important genes, how it prevents fusion of chromosome ends, also how it protects the chromosome ends from degradation. Now, after seeing this, we might wonder that when we have telomerase enzyme, why doesn't this enzyme keep on extending the telomere length after every cell division? That is because in somatic cell, we don't have telomerase activity, and that's why the cell have a finite time of cell division. Once it reaches a critical telomere length, the cell will stop dividing. So where this telomerase activity is found? It is found in embryonic cells, in adult stem cells, and it is also seen in cancer cells. So that's all for now. I hope this video was helpful. Do subscribe to the channel for new video every Friday, and I will see you next time. Until then, keep learning.